Hi everyone, welcome back to my channel. Today I'm going to take you through the transport across cell membrane section of the cells unit for AQA A Level Biology. Also, at the end of the video I'll be going through a few exam style questions and then mark schemes, and as always in the comments section I will be putting timestamps so you can skip to the relevant sections that you wish to revise from. So let's get started. So here I have created myself the basic structure of the cell surface membrane. Now this is the key term that you need to remember. The cell surface membrane is almost always a phospholipid bilayer. So as you can see, I'll just get my pen out here. This here is a phospholipid, this kind of tadpole-like structure if you like. And there are two layers of that, which is why it's called a phospholipid bilayer. The phospholipid bilayer membrane is made up of many components, for example glycoproteins, which are proteins embedded in the membrane but with polysaccharide chains attached to them. And also we have glycolipids, which are polysaccharide chains attached to lipids, so phospholipids. These have many functions, but their main function is to aid in cell attachment. The next things we have are channel proteins and carrier proteins. I will go into their function a bit later on, but their main function is to allow larger or polar or water-soluble molecules across the membrane. Also we have here these little red dots which are dotted throughout the membrane, are called cholesterols, and the role of cholesterol in the membrane is to restrict movement, so to provide stability for extreme conditions. Now the cell surface membrane is known as what we call a fluid-mosaic model. The term fluid here comes from the fact that the molecules in the membrane can flow freely, so to allow flexibility. And the mosaic part comes from the fact that there are many types of molecule in the membrane, for example cholesterols, phospholipids, glycoproteins, etc. etc. So you need to know about a few different types of transport across the membrane. The first example is simple diffusion. Remember that diffusion is the net movement of particles from a high concentration to a low concentration until equilibrium is reached. So simple diffusion is the movement of small, nonpolar or lipid-soluble particles through the phospholipid bilayer. So these molecules just pass straight through the phospholipid bilayer as they are lipid-soluble or small or nonpolar. The next type of diffusion is facilitated diffusion. This is the movement of larger polar or water-soluble molecules through channel or carrier proteins. Now, movement through channel and carrier proteins are different. If something moves through a channel protein, for example water, ions and sugars, for example glucose, you just kind of pass straight through. However, in carrier proteins this is slightly different. So you have your molecule here, for example your sugar or amino acid, and this moves in to the carrier protein. Now what happens here is the carrier protein changes its tertiary structure, so the binding of what we call the substrate, for example the sugar, changes the tertiary structure of the carrier protein, which allows the molecule to be entered into the cell. The next type of transport is active transport, which you will probably remember from previous years. Active transport is movement against the concentration gradient. This means the molecules are moving from a low concentration to a high concentration using carrier proteins requiring ATP hydrolysis. Don't get confused, channel proteins aren't used for active transport, only carrier proteins are. The key term here is requiring ATP hydrolysis. Often the examiners don't accept you just write in requires ATP, often requires you to put ATP hydrolysis, as you will see now. So here is a pretty terribly drawn structure by me of a carrier protein and a molecule being moved by active transport, and as you can see this requires the hydrolysis of ATP. as the hydrolysis of ATP provides energy for the movement against the concentration gradient. This energy comes from activation by the phosphate molecule here that is released during ATP hydrolysis. So the next mode of transport that you need to know about is co-transport. Co-transport is when two molecules are transported together, one going down its concentration gradient and one going against its concentration gradient. Now, the AQA specification wants you to know this in the context of the absorption of glucose via the small intestine, or otherwise known as the ileum. So the first step in this process is sodium ions and glucose, which is denoted by the green G here, are co-transported from the lumen to the epithelium. The lumen is a space inside the small intestine, and the epithelium are cells that line the small intestine. So sodium is going down its concentration gradient and glucose is going against its concentration gradient. So as you can see, they are moving into the epithelium. So the next step is when these sodium ions are actively transported into the blood from the epithelium through carrier proteins. Now at the same time, potassium ion is pumped into the epithelium from the blood, and this process ensures that a favourable concentration gradient of sodium is maintained to ensure that glucose can be absorbed into the blood. So the final step of this whole process is that glucose is transported from the epithelium into the blood via the process of facilitated diffusion through channel proteins. Now glucose is in the blood, it can be transported to various bodily tissues for use in respiration. Now I think this is the last mode of transport that you need to know about, is osmosis. Osmosis is the movement of water molecules, key term here is water, across a selectively permeable membrane from a high water potential to a low water potential. Now, water potential is what I like to remember as a fancy term for water concentration. So it's the relative tendency of water to move from one place to another. So if something has a high water potential, it means that it has a high water concentration, which means that the water molecules are more likely to move to a place where there is a lower water concentration or a lower water potential. For example, in a highly saline solution. So here we have two sides of a membrane with the membrane here in between. As you can see, this side of the membrane, the left-hand side, has lots more water molecules, so it has a higher water concentration. This means that this side, the left side of the membrane, has a high water potential, and this side has a low water potential. This means that the water molecules tend to move from the left side to the right side, as the left side here has a high water potential and the right side has a low water potential. If the water potentials on each side are balanced, we call this isotonic. Now, the last thing that you need to know is some adaptations that cell membranes can have. First thing is an increase in surface area. If there is an increase in surface area, for example, if the membrane is folded, for example, the cristae in mitochondria, it means that more molecules can be transported across the membrane for various cellular functions. Also, there can be an increase in the number of carrier or channel proteins, providing a similar benefit to the increase in surface area as more polar or water-soluble or larger molecules can be transported across the membrane. So that is it for the content. Now we can get on to some exam-style questions. So let's look at this first one. A group of students carried out an investigation to find the water potential of potato tissue. The students were each given a potato and 50 centimeters cubed of a 1 mole per decimeter cubed solution of sucrose. They used a 1 mole per decimeter cubed solution of sucrose to make a series of different concentrations. They cut and weighed discs of potato tissue and left them in the sucrose solutions for a set time. They then removed the discs of potato tissue and reweighed them. The table below shows how one student presented his processed results. So here we have the table that the student obtained. Now this is probably familiar to you, as I think this is the third required practical for AQA A Level Biology. So let's look at the first part of the question. Explain why the data in the table above are described as processed results. Now, the term process means that something has been calculated. And as the heading here says percentage change in mass of potato tissue, then that signifies that something has been calculated. So we can write calculations have been made from the raw data, which is this part. So let's look at the mark scheme. Calculations are made from raw data, so we'll get the mark. Or you can put raw data would have been recorded in initial and final masses, as the student has recorded percentage change. So let's get on to the next question, which is a maths question, everyone's favourite. Okay, describe how you would use a 1 mole per decimeter cubed solution of sucrose to produce 30 centimeters cubed of a 0.15 mole per decimeter cubed solution of sucrose. Now, these questions can be quite tricky and complicated, but I'm going to take you through these now. The first thing that you need to do is you need to find what we call the dilution factor, which is how much something has been diluted. To do this, we divide the original concentration, so this value here, by the end concentration, which is 0.15. So we would get 1 divided by 0.15, which is 6.6. Now I've written the answer here, but I'm going to take you through how I got the answer now. So we've calculated dilution factor. Next thing that we need to do is we need to figure out how much of the sucrose solution we need. To do this, we need to divide the needed volume by the dilution factor, which would get to 1 decimal place 4.5 centimeters cubed of sucrose solution. So we have written that here in our answer. You would add 4.5 centimeters cubed sucrose solution. The next thing, or the final step that we need to do, is we need to make this up to 30 centimeters cubed. So therefore we write 30 minus 4.5. So that means we need 25.5 centimeters cubed distilled water required. Now you would now write this up as a sentence. So you would add 4.5 centimeters cubed sucrose solution to 25.5 centimeters cubed of distilled water, which I've written right here. So let's look at the mark scheme. Add 4.5 centimeters cubed of you don't have to put the concentration, but you can if you want, solution to 25.5 centimeters cubed of distilled water. So you would get both marks for that, as we wrote both of those. Now here it says, if incorrect, allow 1 mark for a solution to water in a proportion. So you can write the proportions instead of actually writing the volumes. But you can only get 1 mark if you write that. So let's look at the next one. Explain the change in mass of potato tissue in the 0.4 mole per decimeter cubed solution of sucrose. Now, as this is an explain question, you need to write why something is happening, not just what is happening. So if we look at the data for 0.4, which is this bottom row here, so as you can see, the change in mass of the potato tissue has decreased. This signifies that water has moved out of the potato tissue by osmosis, signifying that the potato tissue had a higher water potential than the solution. So we can put the water potential of the solution is less than the potato tissue, so the potato tissue loses water by osmosis. Or you can put the water potential of the potato is more than the solution. It doesn't matter. So let's look at the mark scheme. Water potential of the solution is less than, or you can put more negative than, that of the potato tissue. Now it allows this symbol here, which is psi, I think, as equivalent to water potential, as this is the symbol that you can use, but I would recommend just writing it out. So you would get that mark. And tissue loses water by osmosis. The key term here is by osmosis. So we would get these 2 marks. So look at the next question. Describe how you would use the student's results in the table above to find the water potential of the potato tissue. Now you probably would have done this in the third required practical, which is making a calibration curve. Now to do this, You would plot a graph with the concentration on the x-axis and percentage change in mass on the y-axis. Don't get these y-axis and x-axis confused. Then find the concentration where the curve crosses the x-axis. Now, you can't tell what the water potential is from the graph. So you would need to use an external resource to find the water potential of the sucrose concentration where the curve crosses the axes, as you cannot tell just from your own knowledge. So if we look at the mark scheme, mark point 1, plot a graph of concentration on the x-axis and percentage change in mass on the y-axis. Find concentration where the curve crosses the x-axis or where percentage change is 0. Use another resource to find the water potential of sucrose concentration. So we got all 3 marks there. Let's look at the next question. Scientists investigated the percentages of different types of lipid in plasma membranes from different types of cell. The table shows some of their results. So we've got this nice table here. Scientists expressed their results as percentage of lipid in plasma membrane by mass. Explain how they would find these values. So you need to explain how they got the values here. So percentage. You need to divide the mass of each of the lipids by the total mass of all lipids to get the proportion. Now, as we need a percentage, you need to then multiply this by 100. So let's look at the mark scheme. Divide the mass of each lipid by the total mass of all lipids, and you can put in that type of cell, but that is not essential as it is in brackets in the mark scheme, and multiply answer by 100 to get a percentage value. Let's look at the next one. Cholesterol increases the stability of plasma membranes. Cholesterol does this by making membranes less flexible. Suggest one advantage of the different percentage of cholesterol in red blood cells compared to cells lining the ileum, which is the small intestine. Now, as this is a suggest question, it often requires your own knowledge, not that from the specification, just your own ideas. This is what I have put. Red blood cells are free in the blood, so this means that they are not attached to other cells. Now, one advantage of cells being attached to each other is that they can provide stability. However, red blood cells obviously don't have that because they are free in the blood. So this means that they have to have more cholesterol to help them maintain their shape, to provide their function. So let's look at the mark scheme. Red blood cells are free in blood or not supported by other cells, so cholesterol helps to maintain shape, so we would get the mark. Now it says here, allow converse for cell from the ileum. So cells supported by others in the endothelium, so cholesterol has less effect on maintaining shape. So you can write this instead of actually referring to red blood cells. Now I think this is the last question, yes. E. coli has no cholesterol on its cell surface membrane. Despite this, the cell maintains a constant shape. Explain why. Now, as I said in my structure of eukaryotic and prokaryotic cells video, E. coli has a cell wall. And as it is a prokaryote, it is made of murin, not cellulose. As cellulose is a component in a plant cell wall. So this would be 2 marks, as you have mentioned the cell wall and that it is made of murin. Here's the mark scheme. You can write any 2 of these, but you cannot get 3 marks if you write all 3 of these marking points. You can only get a maximum of 2 marks. Here's the first marking point that you could have written, but we didn't write. A cell is unable to change shape. Because the cell has a cell wall, which we wrote. The wall is rigid, or it is made of peptidoglycan or murin. You can write either of these 2, they're basically the same thing. Right, here is the last one. Read the following passage. I'm not going to bother reading the whole thing. You can pause the video if you would like to read it. Now, I'm going to focus on this part of the question as it is relevant to the transport section. LDL enters the liver cells in lines 3 to 4. Now we can focus here on lines 3 to 4. So 1, 2, 3, 4. So lines 3 and lines 4. Here it says liver cells have a receptor on their cell surface membranes that LDL binds to. This leads to LDL entering the cell. Now the question says, using your knowledge of the structure of the cell surface membrane, suggest how LDL enters the cell. Now, the first thing to point out from the text is that LDL binds to a receptor. So we can write that here. And then we can write that it enters through a channel protein, as the receptor gives it an indication that it requires a channel protein to enter the cell. So if we look at the mark scheme, you can actually put that LDL is lipid soluble, and as I said earlier, lipid soluble molecules can enter through the phospholipid bilayer. Or you can put here that the LDL attaches to a receptor. Now, it is crucial that you put that it attaches to the receptor, otherwise you don't get this mark. And that it goes through the channel or carrier protein. You can write either one of these proteins, it doesn't matter. Now here, as it says or, it means that you can't put, for example, it's lipid soluble, so go through a carrier or channel protein. You need to write these 2 points here in pairs. Now let's look at these points here. It says except by facilitated diffusion or active transport, as this is what carrier and channel proteins are used for. Now here it says reject active transport through channel protein, as active transport only happens through carrier proteins, as we mentioned earlier. Now, as it says reject, if you write this here, you don't get any more marks for the question, even if you wrote a marking point that was correct. Right, that is it. That is all I want to say. Please comment below if you have any questions, and I'll see you in the next video.